afternoon and happy Aloha Friday. My name is Whitener.
I'm Landfried.
I'm Emerson.
I'm Hayley.
I'm Annalee.
I'm Sammy, Omar.
And today we will be presenting on Chlordiazepoxide and Clomipramine as effective anxiolytic and antidepressant treatments in rats.
So this current study has a lot of relevance, especially in the field of medicine and mental health. We know that anxiety and depression are two of the most prevalent mental disorders in the United States as well as the global population. And in order for people to receive treatment from drugs specifically, they must be tested through animal models first before moving on to clinical trials and validation by the FDA. So we start with animal models and we use a battery of behavioral testing that tries to see if the drug is something that could even be applicable to humans. And for this study, we use Chlordiazepoxide and Clomipramine, which are two commonly prescribed drugs to treat anxiety and depression.
So as Abby said, we use Chlordiazepoxide and Clomipramine. Chlordiazepoxide is a benzodiazepine, which treats anxiety. It's a GABA agonist, so it works to treat anxiety by inhibiting central nervous activity, which calms anxious behaviors and thoughts. Then we use Clomipramine, or CMI, to treat depression, and that's a tricyclic antidepressant, so it targets serotonin and norepinephrine to decrease depression. So we had a few research questions. Our first one is, does Chlordiazepoxide decrease anxiety in rats? We use the rats as an animal model, as Abby talked about, because we kind of couldn't do the study in humans. And we tested this by giving half of the rats Chlordiazepoxide and half of the rats no drugs, and then testing them on the elevated plus maze, which is a measure of anxiety. Then we also examined ultrasound exposure, which is a new model to induce depression in rats, and this model is environmental stressors. We had speakers over each cage that play a noise that the rats really don't like, that stresses them out. We did this for a period of 28 days, and then we tested whether or not they were depressed using the forced swim test and the sucrose preference test before and after exposure. We also used those tests when we were asking our third research question, does Clomipramine prevent and treat depression in rats? So we gave half of the rats Clomipramine and half of the rats no drug to see if there was a difference after exposure.
So like we mentioned before, we use the elevated plus maze as our model to reduce anxiety in the rat model. What's important to know about the elevated plus maze is that it uses the rat's juxtaposed desire to stay in enclosed spaces, as y'all can see in the closed arms, and their desire to explore novel environments. So in the current study, we hoped that by administering a drug, we could increase time spent on those open arms and entries into open arms to demonstrate an increase in their exploratory behaviors.
For the sucrose preference test, we used a 1% sucrose solution for the first and second test and a 0.5% sucrose solution for the third test in order to account for the ceiling effect found. 2 bottles were placed on the rats' cages. One was filled with the sucrose solution, the other was filled with deionized water. We had a total of a 12-hour exposure time, and bottle positioning was switched halfway throughout exposure. We also did a forced swim test as a measure of depression. For the forced swim test, each rat was placed in a trash can and their behaviors were measured for 10 minutes. We measured immobility, which is the absence of any movement, struggling, which is frantic movement of the forelimbs, particularly along the wall of the trash can, and swimming, which is movement of the forelimbs or hindlimbs in a paddling fashion. After the 10 minutes was up, the rats were placed under heat lamps before returning to their home cages.
So for the elevated plus maze, we expected that the rats that received Chlordiazepoxide would have an increased number of entries onto the open arms and an increased time spent on the open arms, because when rats are anxious, they don't like to be in open spaces with bright light, so the anxious rats would spend more time on closed arms. For ultrasound exposure, we expected that the control group would have a decrease in sucrose preference because they wouldn't be seeking rewards. When you're depressed, you don't normally like to seek out things that are going to make you feel a lot better, like sugar water in this case for rats, and we expected that they would spend more time immobile on the forced swim test because the immobility is them kind of giving up, and when you're depressed, you tend to do that. With Clomipramine, we expected that the drug group would have a higher sucrose preference and spend less time immobile because they wouldn't be as depressed.
When we analyzed the results for the elevated plus maze test, we did find that the rats who had been injected with the chlordiazepoxide prior to completing the elevated plus maze test spent significantly longer amounts of time on the open arms of the maze as compared to our control rats, which can be seen in this graph up top. We also found that those rats who had been injected with chlordiazepoxide showed significantly more entries into the open arms of the elevated plus maze as compared to our control rats, which can be seen in this bottom graph. Both of these findings are supportive of our hypotheses because they suggest that the chlordiazepoxide was effective in reducing anxiety-like behaviors in the rats. When we ran the results for our sucrose preference test, no significant main effects or interactions were found. The rats that were being injected with the clomipramine daily did not show a significantly higher preference for sucrose, which actually contradicted our hypothesis. If you look at this top graph, all of the numbers above 50% suggest a preference for sucrose, and almost all of our rats were relatively close to 100%, which does suggest a ceiling effect for this test. We were also measuring body weight throughout our experiment in order to determine if clomipramine caused a decrease in the body weight of the rats who were being injected with it. However, we found no significant interactions between day of body weight measurements and the groups, with them being control and clomipramine.
So when looking at the forced swim test, we had them swim 3 separate times. And this first graph is comparing the rats before they received any treatment intervention and about 24 hours after their last injection of clomipramine after almost 4 weeks of testing. So we see a main effect of immobility in that CMI reduced immobility in rats for the first minute of the test. And this graph zooms in on the first minute. So you can see on the left-hand side before, all the rats are relatively struggling or climbing, so there's really low immobility. That's showing that they're stressed, they're trying to get out, and they don't have a lot of learned helplessness tendencies yet. However, afterwards, we see that for the control group, their immobility shot up, showing a lot of learned helplessness tendencies, while the clomipramine post-test group, it's still elevated compared to before, but it is lower than the control group. And you can see the inverse when looking at struggling and climbing. This is most salient in the first minute. So just like how clomipramine would reduce immobility, it would also increase struggling. So before any drugs were given, the rats were pretty much struggling and swimming equally, showing that they had the same drive and intentions. However, afterwards, only the clomipramine group really struggled significantly more than the control group. We ran another test looking at the first post-test and the second post-test. Our second post-test was different in that both groups of rats received a singular acute injection of clomipramine. So the control groups who had been neutral for almost 4 weeks received 1 injection, and the CMI rats also received what would be considered like a booster injection, and this was given 1 hour before the test. And we see that the chronic and acute condition for CMI decreased immobility for the first 8 minutes of the test compared to the first post-test. And there were no significant main effects or interactions for struggling or climbing, but you can still see with the dark red bar that it is significantly, I say that lightly, it's taller than the dark blue bar, showing that in the first 2 minutes they did struggle more. And lastly, we compared the pre-test, the first post-test, and the second post-test. And in terms of immobility, we found that the chronic and acute condition was most effective at reducing immobility across all minutes of the trial, specifically when comparing it to the pre-test, the post-test, and the chronic and acute condition.
So for the summary of our results, we found that the chlordiazepoxide injections resulted in increased time spent and entries into the open arms of the elevated plus maze. That showed us 2 things. First of all, it validated the elevated plus maze as a measure for anxiety, and it also showed us that the chlordiazepine is an efficient drug, and it can be efficiently used as an anxiolytic. Furthermore, we've already stated that we found that there was no effect of the clomipramine on the sucrose preference. We think this is less so because of the test and more so actually because of a possible ceiling effect. The rats just specifically like the sucrose solutions much more than the deionized water, so approach that solution much further. Furthermore, for the forced swim test results, we found that after 29 days of ultrasound-induced depression, comparing the control groups and the clomipramine groups, there was a drug effect, but only for the first minute. Specifically, the clomipramine antidepressant reduced the immobility and increased struggle time only for the first minute of the forced swim tests. We think the effect didn't last throughout the entire test, possibly because of general physiology, a possible decrease in energy, increase the immobility, and decrease the struggle times for the rest of the test. Furthermore, something that got us excited was comparing the control group before and after the 29 days of ultrasound exposure showed that there was a significant increase in immobility. So this kind of shows like that factor of learned helplessness, which verifies the efficacy of our ultrasound in inducing depression within the rat models, which is exactly what we want. Then going further, we compared the chronic and acute injections to simply just the acute injections of the clomipramine, and we found that the chronic injection with the acute injection significantly decreased immobility. But this time, instead of just the first minute, it existed for every single minute throughout the test. And this is possibly attributed to the fact that the clomipramine breaks down into its metabolite, desmethylclomipramine, and has a half-life of about 21 hours. The clomipramine acts on the serotonergic system, so it kind of affects serotonin within the brain. And then the desmethylclomipramine acts on the noradrenergic system, affecting norepinephrine. So when you have the chronic injection with the acute injection, it sort of acts synergistically, kind of affecting both those mechanisms in the brain to further reduce immobility for the entire time. Furthermore, comparing the control group before and after just one single acute injection of the clomipramine also showed a significantly reduced immobility, but again, that was only for the first minute of the test. Again, that could be associated with the effects of the drug on the serotonin system within the brain. Future alterations of our study, we think that a short brief access test might be super beneficial instead of a 12-hour sucrose preference test. So once again, we received that ceiling effect, and we think just a short exposure of the rats to the sucrose solution and the deionized water might help in preventing a sort of preference from developing and sort of preventing that ceiling effect. Furthermore, we think shorter intervals between the administration of the clomipramine injections might be beneficial. So instead of administering every 24 hours, since we've already stated that it has a half-life of 21 hours, it would be beneficial to inject in shorter intervals so that you get that combined serotonergic and noradrenergic effect rather than have it completely break down to its metabolite.
Thank y'all for listening. Do you have any questions?
I have a question. What would you say are the implications of your findings?
Our implications build largely on existing literature. So we already knew that these models that we were testing were relatively valid. However, none of them are ever going to be completely perfect for trying to find effective treatments for anxiety and depression, because rat models aren't perfectly translatable to human models. So we were just building on existing literature, and because ultrasound is relatively new, so we were looking and investigating to see if we could do that on a smaller scale or on a less sophisticated scale almost, because we don't have the funding of larger labs.
And the thing with ultrasound exposure is that it more realistically models human depression because it's based off of environmental stressors for rats, when typically research with rats trying to get them depressed previously has been physical stressors such as electric foot shocks. So this will more closely model humans, and hopefully that means that testing the drugs this way will mean that the drugs will be more effective in human trials.
Any questions?